Right. So we are now being recorded. So this is our 10th and final video teleconference for this semester for this subject BIOL 105L section YC. So if we look at our calendar, we are actually doing it ahead of time instead of doing it on Friday because I'm not sure if I can actually be available on that day, not to meet you or to meet with you. So that's why we're doing it earlier. And this is our last video teleconference. So if there are questions, di ba, that you want to ask personally, you may ask them, no, in this video teleconference. So this is on a special topic, no, not any of the laboratory activities, but instead it's from the book, our textbook in lecture chapter 16 on cancer. After this week, next week is already, if you look at the schedule, um, you can actually already use next week to review your other subjects if you have exams. For the final requirement of this subject, I'll be giving them also early next week. So you will have time to do them. And of course, the finals week will be on the second week of May. Okay, so after this week, if you want to, you can already rest, no, for a majority of our time for this subject. Okay, so but we have to talk first about this last topic because this is interesting. And in cell and molecular biology, I think it's going to be incomplete if we won't talk about the abnormalities of cells, and that's in cancer. Okay, so cancer is actually a very scary topic. Okay, I don't know if all of us have encountered cancer on what personal basis ba or ano, someone close to the family experienced this. But this one is a very scary topic, and also interesting at the same time. So we're going to learn at least, no, the key points that we are expected to know about cancer in cell mol. So notice in this particular slide, this is actually the karyogram of a cancer cell. And the karyogram itself already tells you, this is a human cell. But notice instead of 23 pairs of chromosomes, there are abnormal numbers of chromosomes for certain pairs. For example, in chromosome 4, there are 3 of them instead of a pair. And in chromosome 16, there are 5 of them instead of a pair. And chromosome 6, instead of a homologous pair, they're supposed to be identical in shape. They are not. So the abnormalities that we see in this karyogram reflects the abnormality of a cancer cell. And I think it's already understood, by all of us that cancer is something, is a reflection of something wrong in the genetic level, as shown by these chromosomes here. So our learnings on cancer actually accelerated because of the cells that were taken from the cancer. I think it was a breast cancer, no, of Henrietta Lacks. She died in 1951 because of her complications of her cancer. And during that time, no, she was hospitalized. During that time, there was no law yet on taking samples or cells from a patient. So the doctors, when she passed away, actually took the cells, the cancer cells that were on her body. Okay, so although she died in 1951, her cells, which we now call as HeLa cells, are still alive until today. Okay, so By this, we can already say that cancer cells are they have an eternal lifespan. Okay, they are immortal. Okay, as long as you provide them with food and resources, they will continue to divide and live. So these cells are very much old already, no, from 1951 until the present day. In fact, if you study about cancer later on, you can actually order HeLa cells from a bank where they store these cell samples, and you can use them as your laboratory specimen. So these are cancer cells, the ones that are shown here. So, which leads to the question, no, since we talked about cancer and then they are cells, is unicellularity really a lowly condition? Is this a disadvantage to organisms that are unicellular? Okay, so for example, this one here is a paramecium. Okay, being unicellular might uh, tell you, or you might have already have the feeling that if you're unicellular, it's easy for you to get hunted by predators, as shown here. No, so we see here an amoeba eating a paramecium. Okay, so poor paramecium. However, on the other side of the story, if the paramecium actually evades the amoeba, it can actually do a cell division wherein both of the cells are clones of each other, no? A fission, binary fission. So, in a way, if one of these paramecium will survive, it is actually a clone of the other. Now, and as long as one cell survives, the life of the original cell is preserved. So so that there is no ending talaga of that particular lifespan of that organism. So what do you think, guys? Is this a good thing if you are able to clone yourself, and that clone of yours will be able to put that clone in a safe place so that that clone can clone itself again and again and again, and that clone is actually you? Okay, what do you think about that one? Is that something good or bad? Anyone? It's an interesting thing, okay, for unicellular organisms to be able to clone themselves, and the clones are exactly like themselves. Okay, so the question doesn't end here, though. Okay, this leads us to the question: is multicellularity a better solution to survival? Because when you're multicellular, you have the ability to grow bigger, and of course, you have a better chance of fending yourself against predators. Now, so, for example, we have here the sponge, which is the most primitive multicellular animal. Here we have two multicellular animals, the predator, the lion, and the prey. Okay, so the lion seems to be really at a good advantage because it's a predator, it's strong, it has sharp teeth, it can run fast, it's multicellular. We humans also have the advantage of being multicellular because we are able to do things that unicellular organisms cannot. In fact, we are endowed with the ability to think thanks to our nervous system and the neurons. Okay, we are able to create things. We're able to create technology. And if you look at the face of the world right now, it's covered with things that we have created. Okay, so is multicellularity a better solution to survival? We all know, though, that Steve Jobs, as shown here, however brilliant he was with the things that he created, died out of cancer, diba? And cancer, as we've learned a while ago, arises from abnormal cells. Now, so if you're multicellular, you have a problem, and that problem is keeping track of the trillions of cells that you have in your body. Okay? So for example, right now we have trillions of cells. How sure are you that all of those cells are growing normally? Okay? Isn't there one cell there on your skin that's dividing abnormally? I mean, just look at your skin, and if you see moles, those are actually sort of cells, that clumps of cells that have divided abnormally as compared to the surrounding skin cells. No, they might not be cancer, but they're related to cancer. Okay, tumors can become cancerous later on. So that is the price of multicellularity. And what I actually remember here is Jose Rizal. If we connect this thing with what Rizal said of criminals, of corrupt officials in the society, they are what? What did he say? Ang blank, mga blank ng lipunan. What did he say? Do you recall what he said?
Mga kanser ng lipunan.
Yes. Okay. So ang mga corrupt na officials, mga criminals, those who don't abide by the law, and those who harm other people, mga cancer ng lipunan ang kanyang description. And Rizal was a biologist, he was a doctor. He knows about this. And as I told you, if there are trillions of cells or millions of citizens in a country, it's so difficult to keep track who's following the law, who's not. So the same story with the cells in our bodies. We wouldn't know if there's a cell na abnormal ang cell division or all. So that's the price, no, of multicellularity. You want to be multicellular, you have to pay the price of not being able to keep track of all the cells in your body. So here is a picture of someone suffering from cancer. Obviously, no, there's a growth here at the retina, at the back of the eye. This is retinoblastoma. No, and even babies are affected by cancer already. So don't think of cancer as only for the oldies. No, even newborns can have cancer. So why cancer? Because when you view a tissue that is composed of cancerous cells, it looks like it's having arms. So look at these projections that we see here. No, so so people then called it cancer because it looks like a crab, with the many appendages that are projecting from the center. So that's why it's called cancer. But there are actually multicellular organisms like this naked mole rat that they're not only amusing to look at, but scientists have found out that they have a way of evading cancer. By that, I mean that they don't develop cancer cells. And so right now, if you read about this naked mole rat, scientists are studying its genes and how those are controlled so that we'll know what is their secret, bakit hindi sila nagkaka-cancer, even if they are multicellular organisms. Okay, so there might be hope, no, if we unlock the secret of this animal here. Okay, so let's talk about the properties of cancer cells. Number one, they come from normal somatic cells, just like criminals. Criminals come from normal citizens, di ba? So it's so interesting to talk about the parallelism of these things. Okay, they defy regulatory influences. So they do not follow rules. They divide themselves again and again. They do not stay put in place. Okay, they move about. And when cultured in the laboratory, they will continue growing even when the food that has been provided for them is depleted. So they are seemingly immortal. just like what I told you about Henrietta Lacks cells, since 1951, it has still been dividing until now. They are genetically unstable, okay? And their chromosomes, if you have a karyogram of their chromosomes, as what you've seen in the slide a while ago, they exhibit aneuploidy, abnormality in their chromosomes, in their DNA. This one, they are protected from apoptosis. What is apoptosis?
Cell death, sir.
Okay, this is the programmed cell death. And cancer cells, this adds to their power. Okay, they are, at least a majority of them, are resistant to cell death. Okay, can you just imagine this enemy, diba, that's going to be in the body of someone na resistant to death, seemingly immortal, does not follow rules? Who wants this in their body? No one. Okay. Um, they depend on glycolysis for ATP generation. So if you can remember, human cells rely on oxygen, di ba, to do cellular respiration, to make ATP. But cancer cells, they prefer glycolysis instead of cellular respiration. So this is an illustration, the one here on top, of an epithelial tissue. You can see here in yellow, this is the basement membrane. So these are all normal cells here. But then here, we have some cells that have sort of become uncontrolled in their division. So look at them, they are now filling the insides of this tube. Okay? But these cells are still able to stay in place. Okay? So this is what we call a benign tumor, no, benign. Meaning to say, if the doctor removes this from the body through operation, then you can expect to be okay. Okay, so benign tumor. However, if certain mutations are accumulated in a cell, they can actually have the ability to spread to the other parts of the body. So question, in these malignant tumor cells, what enzyme do they use to break through the basement membrane? Do you remember the enzymes used to break the extracellular matrix?
MMP, sir. Ito, matrix metalloproteinases.
All right, so cancer cells are known to produce excess amounts of MMPs. Okay, so here is a micrograph. No, here's a true picture at the bottom. So here on the left, we have normal cells. Then here in the middle, these two pictures show benign tumors. So look at them. They are sort of different from the normal cells that we see here. They're darker in color and they're more clumped, but they stay in place. So you can see the basement membrane is not broken. However, in the last picture here on the right, this one shows cancer cells. How do we know? Because the basement membrane is now breached. No, kay nag-produce naman og MMPs ang mga cancer cells. So once they have breached the basement membrane, they will then have the ability to enter our bloodstream and travel around our body through the bloodstream, and they can lodge into other parts of our bodies and stay there and start a new tumor there. So this process is called metastasis, diba? So that's how cancer cells spread, and that's why they're so difficult to stop. So when, for example, someone is diagnosed with stage 3 or stage 4 cancer, of course, the first treatment is to operate them and to remove the cysts and the tumors, but they need to be observed because there could be other cancer cells that have lodged in the other parts of their bodies. So here is just a drawing showing how cancer cells behave in the Petri dish. So normal cells, when they grow in one layer, no, until they cover the whole surface. But cancer cells, as shown here in red, if the space is already cramped, they will grow upwards. They will form a clump of cells that you see here. So that's how they are in the laboratory. That's how they behave in the Petri plate. Okay, so here is a diagram of a normal cell, the one here on the left, that prefers, it still undergoes glycolysis because glycolysis is sort of the first part of cellular respiration. But then, it prefers oxidative phosphorylation later on, di ba, sa mitochondria, in order to make energy. But cancer cells love glycolysis. And look at this. They do not really prefer cellular respiration. So this is the reason, guys, why if someone is diagnosed with cancer, they are not allowed to eat sugary foods. Why? Because here in glycolysis, the fuel is of course sugar. And if you have cancer and if you eat a lot of sugar, then you're just feeding your cancer cells, and that's what they like because they prefer glycolysis. So here we see a picture of a cancer tissue, and here in the middle, some of the cells have already died, and it doesn't matter to the surrounding cancer cells. They will continue dividing even if the central part has already died. No, necrosis due to anoxic condition, meaning to say lack of oxygen. So that's the reason why in some cancer tumors, no, the middle of the tumor is already infected with pus, ginana na, and the tumor is still continuing to grow because cancer cells don't care if their other cancer cells are dying out of lack of oxygen. Right. What are the causes of cancer? First, of course, we have chemicals that are known to be carcinogenic. So if you've done organic chemistry in the lab, there are many chemicals actually that we use that are carcinogenic. For example, like benzene. Benzene is carcinogenic. I don't know if you've done BIOL 110 general biology 2, wherein there was a chemical used called ninhydrin. Yeah, ninhydrin is poisonous and carcinogenic also. So chemicals. And then, of course, radiation, like gamma rays, X-rays. So that's why you should not expose yourself to X-rays, no? For example, in the hospital, magpa-X-ray ka, like every week na lang magpa-X-ray. That's not allowed because that can actually trigger some of your cells to mutate, no, and to become tumor cells. And not only X-ray, but also ultraviolet rays. So that's why, pag mahilig kayong mag-bike or magpunta sa beach, dapat may sunblock, no, to at least stop the UV from really penetrating your cells in the skin. So UV is actually a significant cause of skin cancer, no, exposure to UV, long exposure to UV. And then we have tumor viruses. Okay? Certain types of tumor viruses, like the human papilloma virus, can cause tumors. So especially in women, women need to be immunized against human papilloma virus, HPV. No? So for those of you who are listening right now, women, make sure naka-immunize na kayo niyan, di ba? HPV, because once this virus actually gets inside of you, it actually affects your cells, especially in the uterus, so that tumors will form, no, and these tumors can be cancerous. And then diet, of course, no. So especially if you consume a lot of sugars, you might not just end up to be diabetic, but you might also be feeding some of the cancer cells that are already starting to live in your body. Okay. Chronic inflammation. So if you experience chronic inflammation, chronic meaning pamatagalan. Now, for example, toothache, and then after a week, naghubag, nagnana, and then you don't want to go to the dentist because you're afraid of the dentist. So you just let it be until nag 1 month na lang, naara gihapon ang hubag. That is actually dangerous, no? That can actually contribute to changes in the cells surrounding the boil, the hubag, and it might cause them to become tumor cells. And of course, most importantly is to realize that cancer is genetic. Okay, it might not run in your bloodline, but it can be caused by these other chemicals and these radiations and viruses. No, so even if wala mo'y kaliwat na namatay sa cancer or na cancer, you still need to be careful with your exposure to the chemicals and radiations. But of course, if it's in your bloodline, you have to be extra careful because we wouldn't know that we already have certain cells in our bodies that are not following instructions. Okay, so cancer is monoclonal. So in this diagram, we see a normal cell in gray becoming a blue cell. What does that mean? That means there's mutation in the cell, no, in the genes of the cell. Okay, and of course, the abnormal cell will divide itself. So look at the blue cells, they increase in number. Okay, and from one of the blue cells, another mutation arises, and you now have the orange cells, and then the orange cells divide, and then from one of the orange cells, here's another cell that has undergone another mutation. The green cells now divide. Okay, and from one of the green cells, you have this cell that has undergone 4 or more mutations, and this is cancer. Now, you don't expect a cell to become a cancer cell with just one change. It's actually an accumulation of different mutations that favor the cell's ability to divide uncontrollably, for example. So, so I hope you understand this one. No? It's the changes, the accumulated changes in the genetic information of the cell. This is just another illustration. So look at the cell. No? Normal is gray. The gray becomes yellow-orange, meaning to say 1 mutation. And out of the yellow-orange, there's an orange one, meaning to say 2 mutations. And out of the orange one, there's a red one, meaning to say 3 mutations. And the red one is able to produce excess amounts of MMPs. So look at this. It has now broken through the basement membrane. Okay, so you have cancer cells now. So from normal chromosomes, you have this exaggeratedly abnormal karyogram that we see here. Okay, because these are accumulated changes. So any questions so far? If you're curious about a thing, if you have something that you want to ask about our topic, please ask. None? Okay, so we'll continue. Okay, so this time, let's talk about the genes. There are only 2 types of genes that are implicated, meaning to say, pag may cancer, usually sila ang sadan. Sila ang mga genes na naisala. So there are 2 types of them. The first one are the tumor suppressor genes. What are tumor suppressor genes? Can anyone tell us? Or can anyone guess? A guess is okay. Unsa man ang tumor suppressor gene?
Guess lang, sir. Perhaps they help in the regulation of tumor growth within the body, sir.
Okay, so yeah, that's correct. Now they help in the regulation of cell division. So gina-control nila ang number of cell division in a particular cell. So you're correct. So in that sense, they are good when they are working properly. Okay? They are okay. But of course, if we talk about cancer, we're talking about mutations, changes in the genes. So if these genes, which are supposed to suppress tumor, lose their function because of mutation, then you can expect bad things to happen. So here's an example. No? So for example, here we have a pair of tumor suppressor genes, okay, because we are diploid. So notice here in the second division, one of the daughter cells has accumulated a mutation in a pair, one pair. The other pair is okay. Now, in tumor suppressor genes, as long as one pair is still okay, you would still be okay. However, if both of the tumor suppressor genes are affected by the mutation so that they lose their function, then that's the time that that cell loses out of control in the way it divides. Then you can have, it's possible to have a cancer cell here. No, only when two of the tumor suppressor genes, both of them, are affected. Then the second type of gene is the proto-oncogene. Okay. Any guess what a proto-oncogene is? The study of cancer is called oncology. So unsa man yung proto-oncogene? Unsa man ang proto-oncogene, guys? Any guess? Guess lang gihapon, will be fine. Kung ang tumor suppressor genes stop tumor, what are proto-oncogenes? This is much more difficult to guess. So, okay, sige. I'll show you the illustration though. Okay, so ang proto-oncogene, these are genes that have certain roles in the promotion of cell division. So, sort of the opposite of the tumor suppressor gene, no? Ang tumor suppressor gene, ang iyang original role is to control and to stop cell division. Ang proto-oncogene would usually have the opposite normal role, and that's to promote cell division. Okay, so the mutation, the duplication or loss of regulation, meaning to say, nagpakawala ang proto-oncogene, meaning to say, it becomes lenient, okay, and it promotes more cell division, then that's the time it can cause abnormal cells, okay, as shown here. So look at this, the difference between the two. It only takes one pair of a proto-oncogene to mutate before this pair can actually result to a tumor cell or loss of growth control. Okay, so that's the difference between them. In the tumor suppressor gene, as long as one is still okay, that can still override cell division, uncontrolled cell division. But in a proto-oncogene, if one of them is not okay, then they can actually contribute to the loss of control of the cell division. Okay? So in a way, you might say na mas hadlok ang proto-oncogene na mag-mutate. Okay, it only takes one pair and you can have a tumor already as a result. Okay, so that's the difference between them. So once the proto-oncogene has mutated and has become a contributor to the loss of growth control, it is now called an oncogene. So ang oncogene is a cancer gene. Okay, so mao na ang gitawag sa proto-oncogene because it's like the precursor to the cancer gene. Okay, so let's look at specific examples. So genes are actually named with capital letters that are italicized, such as this one, RB gene. Okay. And genes are also named after the disease that happens when they are not non-functional. No? So for example, ang RB gene, this gene, if it's non-functional, can actually cause retinoblastoma. So mao nang tawag sa RB gene. Okay? This is a tumor suppressor gene. So in the general population, retinoblastoma is rare, very rare in the general population. But in certain families, even younger children in certain families can have retinoblastoma, and there's 90% incidence. So from this information alone, can we say that retinoblastoma is genetic or not?
Yes, sir. Most likely it is genetic, sir.
That's correct, because it's observed to be in greater incidence in just certain families. Okay, so most likely genetic, must be inherited. So let's look at the story behind this. Because it is a tumor suppressor gene, in the general population, no, the general population are actually populations with um, two normal RB genes. So meaning to say the genes are working properly, there is no tumor formation. Okay, so let's say in their lifetime they will get one mutation of those pair of genes, RB genes. Will they have retinoblastoma already or not yet? Anyone?
Not yet, sir.
Not yet, diba? So this mutation is actually also quite rare, no? So maybe once in a lifetime. So pag isang mutation lang, only one gene is mutated, you can still live your life normally, no retinoblastoma for the general population. Only when both have mutated would someone have retinoblastoma. And usually that happens in the later stages of life, kanang tigulang na. 'Di ba kay rare man kay ang mutation. So, morning very rare in the general population. But in certain families, what if si mama married papa and both of them have already half of their RB genes mutated? Morning certain families. So when they marry, when they have an offspring, most likely their offspring will receive one half possibility na both of his or her genes are abnormal, mutated, and one half possibility na hindi mutated, something like that. So as a result, in certain families, it is easier for retinoblastoma to happen in the family members. In fact, even at a very young age. Okay? So this is the story behind retinoblastoma, which is a tumor suppressor gene, RB gene rather. Okay. Any questions? Okay, none. So Let's look at the specifics of the function of the RB gene. So we have here again transcription, okay, the making of messenger RNA to make a protein, and this protein will facilitate a cell from G1 to become an S-phase cell. So meaning to say, ang iyang G1 nga roadblock nga checkpoint is it's allowed to go through the checkpoint to become S-phase. Okay? And what the RB gene does is it produces a protein called PRB, which controls the transcription of this messenger RNA. Okay. So when the RB gene is mutated, or if it doesn't function, then your RB protein will also not function properly. And therefore, the transcription of this messenger RNA will continue on and on and on. It will not stop. Okay, wala man si protein RB because your RB gene is non-functional. And so if this will not stop, you will continue making this protein, which allows the cell to continually go from G1 to S again and again and again. So the cell divides much more uncontrollably because of your defective RB gene. Okay, so that's the RB gene story. Next, we have the TP53 gene. So notice the name, all capital letters and italicized, meaning to say that's a gene. This is another tumor suppressor gene. Okay, what is this TP53? Okay, the TP53 gene produces a protein called P53. And this P53 that we see here in teal, and we see here the DNA, is actually known as the guardian of your genome. Okay, it's the guardian of the genome. Why? Because it helps in the repair of damaged DNA, kaning P53. So in fact, in many cancer cells, when scientists study the genes of cancer cells, they found out that all of them would contain mutations in their TP53 gene. Okay, so ana ka-importante ang TP53 gene, a tumor suppressor gene. So what's its function? So here we see a cell with normal TP53 gene. So there are no mutations in its chromosome, so it can continue dividing normally. Okay. Then here we see a cell that is exposed to radiation, for example, and so the DNA in the nucleus is damaged. Okay. The P53 actually helps in the detection of the damage and in the repair of the damage, so that the cell can continue dividing and the resulting cells are normal. No, helps in the repair of the damage. Or the P53 can also instruct the cell, if it's beyond repair, to undergo cell death, apoptosis. So that's the function of your P53. No, repairs the DNA for normal cell division, or tells the cell to just die because it's beyond repair. That's the P53. However, if the P53 is non-functional because your TP53 gene is mutated, then look what happens here. Even if there's damage in the DNA of the cell, it will continue to divide into even more abnormal daughter cells. And the daughter cells will, of course, continue dividing on and on and on. So you have a tumor as a result. So that's your TP53 gene. So remember this diagram for our previous lesson in lecture. Where is your P53? It's here. Okay? And the ultimate goal here is to prevent the cyclin from binding to the cyclin-dependent kinase so that cell cycle is arrested. Okay, so that's the P53 function, no? When it detects damage in the DNA as shown here. Any questions? Kaning TP53 no, nag-study din ng mga scientists, chemotherapy. How is chemotherapy, how is it, is it good? Does it function? So here we have on the vertical column, we have the TP53 alleles. So in the first row, they are both normal. So here on the horizontal axis, you have here the untreated cells, cells treated with different types of chemotherapy. No, if you don't know what chemotherapy is, it's used to kill cancer cells. Okay, so look what happens. If both the TP53 alleles are okay, once a chemical is applied onto the cells, they die. No, so if these are cancer cells and their TP53 alleles are both okay, then they can still be treated with chemotherapy. Pwede pa. But notice this in the cell wherein one of the alleles for TP53 is non-functional. Even when chemotherapy is applied, some cells still survive. Okay? And if both of the TP53 alleles are non-functional, there is no significant difference from before chemotherapy and after chemotherapy. So that's why before patients must decide, no, it's very important to at least know if my cancer cells have functional TP53 genes or not. Because if they don't, then the chemotherapy will just be doing more harm than good to me. But the trouble is, how do you know if your TP53 alleles are all normal? Diba? So kailangan pa ng genetic sequencing and etc. in order to find that out. And even then, there are millions of cells in your body to check. So just saying no, that when you undergo chemotherapy, it's not really 100% effective because of these TP53 allele mutations. So we also have the gene implicated in breast cancer known as BRCA gene, which is a tumor suppressor gene that helps in the repair of DNA damage. Okay, so I'll just go through that. And then oncogenes. Okay, so oncogenes. So oncogenes only require one of the alleles to mutate before it can cause loss of control of cell division. And there are many types of genes that are oncogenes or proto-oncogenes. First, we have the genes that make the growth factors, diba? The genes that make these. What if the gene that makes this is mutated and the growth factor that is produced, once it sticks to the receptor, it never leaves. It just stays there. Okay, dili naman siya matangtang, okay? It has lost that function. So as a result, because this is a growth factor, if it's always there, it will always tell the cell to divide. So you have an oncogene. Another example is this RAS protein. Okay, this RAS protein is has a big role in cell division, and if the gene that makes the RAS protein is mutated and the RAS protein is non-functional, it can actually cause continuous cell division in the cell, of the cell. Okay, what are the other genes? The genes that, let me see, that produce the cyclins. Okay, if the genes that produce the cyclins are mutated, and the cyclin will gain the ability to never leave the cyclin-dependent kinase, then you know the story. The cell will always do cell division no matter what. Okay, so these are the oncogenes, no? And these are the proto-oncogenes. Kaning nasa illustration. Okay, so those are the two types of genes that are implicated in cancer. Number one, tumor suppressor genes. And number 2, the proto-oncogenes. And then there are even certain individuals wherein there are so many mutations in their genes. Now, this is, I think, by chance, of course, by chance or by inheritance, no? Wherein they have so many mutations in their DNA that it's very rampant in their body to have cancer cells, no? And this is what we call as the mutator phenotype, wherein they are at a very high risk of developing cancer. So what are the strategies in combating cancer? There's immunotherapy. Okay, you might want to read about this. We cannot detail it here anymore. There's also the inhibition of the activity of cancer-promoting proteins. There's targeting of the right cells. So definitely when you do chemotherapy, most of the time you are not targeting the right cells. You are also targeting your own cells. Mo nang malagas ang buhok or mag-diarrhea, 'di ba? Kung mag-chemotherapy ka, you are actually also killing your own functional cells. So we need to be able to target the right cells. And lastly, inhibit the formation of blood vessels. Why? Because cancer cells need blood supply all the time. So if we find a way to remove this connection between the blood vessel and the cancer cells, then we can deprive them of nutrients and we can kill them. Okay, so these are just some strategies in combating cancer. Okay, so that ends my presentation on cancer. And if you read the book, there are a lot more details that are interesting that you'll find on cancer. Okay, so are there any questions? Were you able to see my PowerPoint a while ago?
Yes, sir.
Okay, okay. In my view here, it says that I need to present now. Let me see. So were you able to see up to this slide, this one?
Yes, sir.
All right,
Yes, sir.
that's good. Okay, so it's very interesting, no? And in fact, if you're going to be, if you'll decide later on to specialize in the study of cancer, no, I will not be surprised because it's a very interesting thing to be able to find the solution to this, no, or to know how cancer cells work, and of course to be able to help people who have cancer. So have fun reading on chapter 16 about cancer. And if you have any questions, you can still ask them now. Are there any other questions? None? So this is our last video teleconference. All right. So if you need to communicate again, that will be through an online message now. Wala na tayong next video teleconference. So you send your message or questions through eLearn or through email or through Messenger if you have any other concerns. So thank you very much, guys, for joining in this video teleconference. A recording of this will be posted later on so that you can review what we've talked about in this video teleconference. So have a productive day ahead. Enjoy finishing your semester, and you are now all dismissed.
Thank you, guys, sir.
Yes,
All
sir.
Thank you,
right,
sir.
you're welcome.
Thank you, sir.